amoebic dysentery. Shelled saccharidines present a variety of interesting morphological forms, the best known of the shelled forms of the Forma infera. Forma infera are exclusively marine organisms living primarily coastal waters, and the shells called test of different species shows distinctive characteristics and are often quite ornate. Tests are usually made of calcium carbonate. The cell is not firmly attached to the test, and the amoeba cell may extend part way out of the shell during feeding. However, because of the weight of the test, the cell usually sinks to the bottom, and it is thought that the organisms feed on particulate deposits of sediments, primarily bacteria and detritus. The shells of Foraminifera are relatively resistant to decay and hence readily become fossilized. The white cliffs of Dover, England are composed to a great extent of Formifera shells. Because of the excellent fossil record these organisms leave, we have a better idea of their distribution through geological time than for virtually any other protozoa. Ciliates, the ciliates. Ciliates are those protozoa that in some stage of their life cycle possess cilia. They are also unique among protozoa in having two kinds of nuclei. The micronucleus, which is concerned only with inheritance and sexual reproduction, and the macronucleus, which is involved only in the production of messenger ribonucleic acid for various aspects of cell growth and function. Probably the best known and most widely distributed of the ciliates are those of the genus Paramecium, which will be used here as an example of the group. Most ciliates obtain their food by ingesting particulate materials through a distinct oral region or mouth connected to an underlying gullet. Once inside, the food particles are carried down the gullet and into the cytoplasm, where they are enclosed in a food vacuole, a structure into which digestive enzymes are secreted in addition to cilia, which function in motility. Many ciliates have Trico Cyst, which are long, thin filaments of the contractile nature anchored beneath the surface of the outer cell layer. These enables the protozoa to attach to a surface and can aid in defense by signaling the cell that it is being attacked by a predator, thus stimulating cellular defenses. In the case of predatory ciliates, such as Didenium, Trico Cyst holds on to and paralyzes the prey as a prelude to ingestion. Many Paramecium species, as well as many other protozoa, are hosts for endosymbiotic bacteria living in the cytoplasm of the micronucleus. In some cases, evidence exists that these endosymbionts play a nutritional role for the host, synthesizing vitamins or other growth factors that would otherwise have to be obtained from the environment. In the case of protozoa existing in the termite gut, we previously discussed how endosymbiotic methanogens remove H2 produced from pyruvate oxidation in the hydrogenism, yielding CH4, which is released to the atmosphere. Although a few ciliates are parasitic, for animals this mode of existence is less extensively developed in the ciliates than it is in other groups of protozoa. These species, Valentidium coli, is primarily a parasite of domestic animals, but occasionally it infects the intestinal tract of humans, producing intestinal dysentery, symptoms similar to those caused by Entamoeba histolytic. In addition, there is usually a characteristic fauna of obliquely anaerobic ciliates in the rumen, the fourth stomach of ruminants animals. These protozoa play a beneficial role in the digestive and fermentative processes that occur there. The Apicomplexa, or sporozoas, comprise a large group of protozoa, all of which are oblique parasites. They are characterized by a lack of motile adult stages and by a nutritional mode of life in which food is absorbed in solute form through the outer wall, such as occurs in prokaryotes and in fungi. Although the name sporozoa implies the formation of spores, these organisms do not form true resting spores, like those of bacteria, algae, and fungi, but instead produce analogous structures called sporocytes, which are involved in transmission to a new host. Numerous kinds of vertebrates and invertebrates are host to sporozoa, and in some causes an alteration of host takes place, with some stages of the life cycle occurring in one host and some in another. The most important members of the sporozoa are the Coccidia, usually parasites of birds, and the Plasmodia, malaria parasites, which infect birds and mammals including humans, because malaria is a major disease of humans, especially in developing countries. We devote a considerable discussion to this disease and the properties of malaria parasites. Key genera: Penicillin, Aspergillus, Saccharomyces, Candida. In contrast to the protozoa, fungi contain cell walls and produce spores, among many other differences, and most describe species from a relatively tight phylogenetic cluster. Three major groups of fungi are recognized: the molds and yeast, and the mushrooms. This inhabits the fungi are quite diverse. Some are aquatic, living primarily in fresh water, and a few marine fungi are also known. Most fungi, however, have a terrestrial habitats in soil or a dead plant matter, and these types often play crucial roles in the mineralization of organic carbon in nature. A large number of fungi are parasites of terrestrial plants. Indeed, fungi cause the majority of economically significant disease of crop plants. A few fungi are parasitic on animals, including humans, although in general, fungi are less significant as animal pathogens that are bacteria and viruses. For a discussion of pathogenic fungi. Cell walls and metabolism. Fungi cell walls resemble plant cell walls architecturally but not chemically. Although cellulose is present in the walls of certain fungi, most fungi have non-cellulosic walls. Chitin, polymer of glucose derivative and acetylglucosamine, is a common constituted of fungal cell walls. It is laid down in microfibrillar bundles like cellulose. Other glucans, such as mannans, galactosins, and chitosins, replace chitin in some fungal cell walls. Fungal cell walls are generally 80 to 90% polysaccharide, with proteins, lipids, polyphosphates, and inorganic ions making up a self-cementing matrix. An understanding of fungal cell wall chemistry is important because of the existence of biotechnological uses of fungi, and because the chemical nature of the fungal cell wall has been useful in classifying fungi for research and industrial purposes. Fungi are chemoorganotrophs and generally have simple nutritional requirements. Many species can grow at environmental extremes of low pH or high temperature, up to 60 degrees Celsius, and this, coupled with the ubiquitous nature of fungal spores, making the organisms common contaminants of food products, microbial culture media, and most surfaces. Molds and yeast are not, however, classified on physiological grounds, but instead by their diverse array of life cycle patterns, including the formation of a variety of differential sexual spores. Filamentous fungi molds. The molds of filamentous fungi. They are widespread in nature and are commonly seen on stale bread, cheese, or fruit. Each filament grows mainly at the tip by extension of the terminal cell. A single filament is called hypha, plural hypha. Hypha usually grows together across the surface and forms compact tufts collectively called mycelium, which can be seen easily without a microscope. The mycelium arises when the individual hypha form branches as they grow, and these branches intertwine, resulting in the compact mat. In most cases, the vegetative cell of the fungi hypha contains more than one nucleus. Often hundreds of nuclei are present. Thus, a typical hypha is a nucleated tube containing a cytoplasm. From the fungal mycelium, other hyphal branches may reach up into the air above the surface, and on these aerial branches, spores called Canadia are formed. Canadia are asexual spores. The formation does not involve the fusion of gametes, often highly pigmented and resistant to drying, and function in the dispersal of the fungus to new habitats. When Canadia from the white color of the mycelium changes, taking on the color of the Canadia, which may be black, blue, green, red, yellow, or brown. The presence of these spores gives the mycelia mat a rather dusty appearance. Some molds also produce sexual spores formed as the result of sexual reproduction. The latter occur from the fusion either of unicellular gametes or specialized hypha of gametangia. Alternatively, sexual spores can originate from the fusion of two haploid cells to yield a diploid cell, which then undergoes meiosis, a mitosis to yield individual spores depending on the group to which a particular fungus belongs. Different types of asexual spores are produced. Spores formed within an enclosed sac, ascus, are called ascospores, and those produced on the ends of the club-shaped structures, basidium, are basidium spores. Zygospores produced by the zygomictose fungi like common bread molds, Rhizopus are Rhizopus and macroscopically visible structures and the result of the fusion of hypha and genetic exchange. Eventually, the zygospore matures and produces asexual spores that are dispersed by air and germinate to form new fungal mycelia. Sexual spores of fungi are usually resistant to dyeing, heating, freezing, and some chemical agents. However, fungal sexual spores are not as resistant to heat as are bacteria endospores. Either an asexual or a sexual spore of fungus can germinate and develop into a new hypha and mycelium. A major ecological activity of many fungal species, especially members of the Basidiomycetes, is the decomposition of wood, paper, cloth, and other products derived from natural sources. Basidiomycetes that attack these products are able to use cellular or lignin from the product as carbon and energy sources. Lignin is a complex polymer in which the building blocks are phenolic